plant varieties that were developed as a result of this conventional plant breeding. Okay, as a result of this conventional plant breeding. Okay, that is. So when you observe here, India is mainly an agricultural land. India is mainly an agricultural land. So India is mainly an agricultural country. Okay, and next, agriculture accounts for approximately 33% of the India's GDP. So that is a gross domestic product. So 33% of economy is obtained from the agriculture only, and employs nearly 62% of the population. That means 62% of the people, okay, that are present in India are depending upon this agriculture, depending upon the agriculture. And next, after India's independence, one of the main challenges facing the country was that of producing enough food for the increasing population. So after independence, we have observed there is an enormous increase in the population. So okay, so the India is facing certain challenges to meet the demand for the food for this ever increasing population. Okay, so as only limited land is fit for cultivation. Okay, because most of them, okay, they are hilly regions. Okay, and next, forest cover, and next, construction of dams. Okay, because of all this, a lot of land area was not used for cultivation. Only a small area is present for cultivation. So because, okay, that means only a small land area is available for agriculture purpose. So that's why we need to develop some new plant varieties to increase the food production. So the development of several high yielding varieties of wheat and rice in the mid 1960s, as a result of various plant breeding techniques, led to dramatic increase in food production in our country. So that means, okay, during the period of 1960 to 70, 1960s to 1970s, in between this period of 10 years, okay, the food production was increased enormously because of the introduction of new plant varieties into the agriculture. As a result, the food production was increased enormously. This phase is often referred to as a green revolution. Okay, this period of 10 years during which the food production was increased enormously due to the development of new plant varieties. Okay, in that small available land area only. Okay, of increased food production due to the introduction of different plant varieties of rice and wheat. We are calling it as a green revolution. Okay, here we can see, okay, the figure 9.3 represents some Indian hybrid crops of high yielding varieties. Okay, maize variety, and next, okay, wheat variety, okay, beans variety, okay, garden peas. Okay, like this, we used to develop some new varieties. Okay, we used to develop some new varieties. Okay, that is. And next, what are the different plant varieties? We'll discuss one by one. Okay, first we'll go with the wheat and rice. What are the new varieties of wheat and rice that were developed during this green revolution? So, okay, it was started in the year 1960 and it is continuing because every time we are introducing, developing some new varieties and we are trying to increase the food production. So, it was started in the year 1960. Okay, most probably for 10 years we have done, and later on the same continuing almost. So, okay, during the period 1960 to 2000, okay, that is within this 40 years, wheat production increased from 11 million tons to 75 million tons, while okay, rice production went up from 35 million tons to 9.5 million tons. That means okay, within the span of 40 years, okay, the food production, okay, the food grains production was increased enormously. Okay, the wheat production was okay, here we are discussing related to 1 year. In 1 year, they used to produce 11 tons before 1960. Okay, slowly we have improved by the time, by the year 2000. How much wheat grains are produced in 1 year means around 75 million tons. So that means, okay, almost the wheat production was increased by 7 times. Okay, by 7 folds. Okay, the production increased by, okay, the wheat production was increased by 7 folds, almost 7 folds. And at the same time, when you observe the rice production was okay, increased from 35 million tons to 89.5 million tons. That is almost 3 times, nearly 3 times. Okay, nearly 3 times or 2.5 to 3 times. Okay, the rice production was increased. Okay, you know what is the reason? This was due to the development of semi-dwarf varieties of wheat and rice. Okay, this is all achieved. Okay, when you observe here, okay, because of the development of this is or production of production or development of this semi-dwarf varieties, what happens? The lifespan of the plant, okay, the rice and wheat has been reduced. The crop production, okay, the produce for one season has been increased. Before okay, it used to take 8 to 10 months to complete one season. So we used to have only one crop in 1 year. We used to have only one crop in 1 year. But now, because of the development of this semi-dwarf varieties, okay, the lifespan of the plant was reduced to 3 to 4 months. Okay, was reduced to 3 to 4 months. So we can have at least 3 seasons in 1 year to grow this our wheat varieties. So that means we can raise 3 crops in 1 year, and at the same time the production also increased in one season. As a result, overall the production was improved because of the development of this semi-dwarf varieties. So that semi-dwarf varieties were developed. Okay, this is all. Okay, this increase in the food production or food grain production was mainly due to the introduction of semi-dwarf varieties of wheat and rice. Okay, who initially developed this semi-dwarf varieties? Okay, a Nobel Laureate, that means a person who won the Nobel Prize for this development of this semi-dwarf or dwarf varieties of rice and wheat. Okay, he was awarded the Nobel Prize. So that's why we are saying Nobel Laureate Norman E. Borlaug. But because why he was ever given this for science? Okay, for development in science, he was awarded this Nobel Prize. Okay, to this Norman E. Borlaug, because he saved the life of a large number of people by increasing the food production. That's why he was given this Nobel Prize. So Nobel laureate, who okay, that is Norman E. Borlaug at International Center for Wheat and Maize Improvement in Mexico. Okay, it belongs to America, USA. Okay, it belongs to USA. Okay, in that a country called Mexico. Okay, in that okay, research center is there, which is named as International Center for Wheat and Maize Improvement. Okay, it is present in Mexico. He is a person who developed this semi-dwarf wheat, semi-dwarf variety of wheat. Okay, in the year 1940s, 1950s, he has developed. Later, they were brought to India in the year 1960s, and by crossing this okay, this foreign varieties with our local variety, they developed a large number of different types of semi-dwarf varieties which can grow in our local areas. So in the year 1963, several varieties such as Sonalika and Kalyan Sona, which were high yielding and were introduced all over the wheat growing belt of India. Generally, the wheat is grown as a crop in northern parts of India. In most of the northern parts of India, wheat is grown as a crop. Okay, like rice in southern part of India, they used to grow wheat. So in that places, they introduced this new varieties like Sonalika and Kalyan Sona. We can see here. Okay, in the pictures we have given Sonalika, Sonalika wheat variety. Okay, Sonalika wheat variety. In the year 1967, a new variety. In that year, a new variety were developed and what introduced, that is a HD1553. Okay, that is the variety that is given to this name in the year. Okay, we can see here. Okay, every crop field, whichever they improved, okay, every crop field will be placed. If you go to any research areas or any okay, or even the seed companies, okay, crop fields, okay, or the scientific fields, okay, we can see here every crop, okay, field will be having the name board, the variety in which year it was developed. So here we can see, okay, it is a Sonalika variety of HD1553, which was developed in the year 1967. Okay, there are some other new, okay, some other varieties started in the year 1963. Finally, they developed this one in the year 1967. So that's what we can see here. Okay, that is the new variety called Sonalika and Kalyan Sona of wheat or semi-dwarf varieties of wheat. Okay, were developed and they're introduced into the different areas of India where wheat is grown. And later, okay, the semi-dwarf rice varieties were derived from IR8. Okay, that is developed at International Rice Research Institute, which is present at Philippines. At the time, who is the head of this organization means M.S. Swaminathan. Okay, he worked as the head of that organization during the period of 1960s, and he only developed this IR8 variety of semi-dwarf variety of rice. Okay, that is developed at International Rice Research Institute, which is present at Philippines. And next, okay, not only IR8, they developed some other variety like a Taichung native one. Okay, which is okay, that means they obtained a new variety of rice from Taiwan, and it is crossed with our local variety, and they developed a new semi-dwarf variety of rice called Taiwan native one. Okay, sorry, Taichung native one. Okay, these derivatives were introduced in 1966. Okay, in India, okay, they were introduced to India in the year 1966. Okay, and okay, these are the first okay semi-dwarf rice varieties that were introduced into India. That is IR8 and Taichung native one. Okay, from Taiwan, and later the derivatives were introduced in 19. Later, better yielding semi-dwarf varieties like Jaya and Ratna were developed in India. Okay, remember, okay, we have to remember these names very clearly. Okay, they are asking so many times in the examination in 2 marks. Okay, they will give you the variety and they will miss. Okay, they give the crop name and they miss the variety, and you have to identify like A, B, C, D. They'll miss A, B, C, D, and they will ask you what is A, B, C, D. They'll give rice. What are okay? What is the rice variety? Jaya, Ratna, IR8, or Taichung native one. If they give you wheat, Sonalika and Kalyan Sona. Sonalika and Kalyan Sona are the wheat semi-dwarf varieties of wheat, which were introduced in the year 1963. Later, in the year 1966, the semi-dwarf varieties of rice were introduced. First one is IR8, and later Taichung native one. And after this, a lot of different semi-dwarf varieties of rice were introduced, like Jaya and Ratna. Okay. Okay, these are the semi-dwarf varieties that were developed related to wheat and rice. And they also developed some new variety of sugarcane. Okay, sugarcane. Okay, there are two major varieties of sugarcane in India. One will grow in North India and one will grow in South India. Okay, the sugarcane variety that grows in South India is a Saccharum officinarum. Okay, Saccharum officinarum, the sugarcane variety that grows in a southern part of India, whereas Saccharum barberry is another sugarcane variety which grows in a northern part of India. What is the difference here means? Okay, when you observe the northern variety, that is Saccharum barberry, the stem is thin and low sugar content. but able to grow properly, okay, able to grow in northern soils, soils of northern part of India. But when you observe the Saccharum officinarum, which is usually grown in the southern soils of southern part of India, when you observe this variety, the thick is, okay, the stem is thick and more sugar content. Okay, the stem is thick and more sugar content. Okay, it is rich in sugar. So when they, okay, when the, okay, after observing this, okay, Saccharum officinarum in southern part, which is growing, this is, okay, Saccharum officinarum, they introduced this Saccharum officinarum into southern part of India also, but it was unable to grow properly. Okay, it is unable to grow properly because, okay, because of the environmental conditions and all that, or diseases that are shown, okay, that are observed in that area, or the type of soil. So because of so many reasons, this Saccharum officinarum is unable to grow properly, and the yield is also less because it is unable to grow. So what they have done means, they crossed the Saccharum officinarum with that of Saccharum barberi. That means the southern, okay, the southern variety was crossed with the northern variety of sugarcane, and they developed a new variety of sugarcane, which has been successfully grown in northern part of India with a thick stem and more sugar content, and introduced into different places of northern part of India, so that the sugarcane production has been increased. Now India stands second in the production of sugarcane. India stands second in the production of sugarcane. First one is Cuba. Second one is India. We are now exporting the sugar to other countries also because of this increased, okay, because of the increase in the production, because of the introduction of new varieties of the sugarcane. Okay, that is what we can see here. So Saccharum barberi was originally grown in North India, but had a poor sugar content and yield. Okay, it is having thin stem, so poor sugar content and yield, because thin stem means less yield only. But the tropical canes, that is the, okay, the tropical, okay, that is the sugar cane which is grown in a southern part, that is the tropical canes, sugar canes grown in South India. Saccharum officinarum has the thicker stems and a higher sugar content, did not grow well in North India. But when they introduced this Saccharum officinarum to North India, but it was unable to grow properly, and the yield is also reduced. So what they have done means these two species were successfully crossed to get sugarcane varieties combining the desirable qualities of high yield, thick stems, high sugar, and ability to grow in the sugarcane areas of North India. Okay. So by crossing these two, Saccharum officinarum and Saccharum barberi, they developed a new variety, okay, which has the desirable characteristics of both the plants. That is high yield, thick stems, high sugar, and ability to grow in areas of North India where sugarcane is grown. Okay, that is. Okay, and next, like that, the other plant varieties, other crop varieties are also improved. Okay, comes to millets like a hybrid maize. Okay, remember ma, maize. Okay, hybrid maize, jowar, and bajra. Okay, these are the millets. Maize, jowar, and bajra have been successfully developed in India. Okay, generally we can see no, jowar and bajra are usually grown in the tropical regions where water content is less available. Usually, okay, before this bajra and jowar, maize, they are used to grow in the areas where water is sufficiently available. But with a successful breeding program, okay, we have developed some new varieties of these millets, which can grow even in drought conditions. So that's why, okay, that's what we have developed. Okay, generally the maize, jowar, and bajra are grown in the areas where water content is less. Okay, in the tropical regions in India. Okay, that is maize, jowar, and bajra have been successfully developed in India. And next, hybrid breeding have led to the development of several high-yielding varieties resistant to water stress. Okay, these are the varieties. Okay, which are developed in India, that is related to maize, bajra, and jowar. Okay, which are able to grow in a drought condition. That means a water stress condition. Water levels are very less, our water content is less, our moisture in the atmosphere is less, are showing a dry conditions in that area. Okay, yielding varieties resistant to water stress. Okay, like that. Okay, we have developed some new varieties of food grains by using this plant breeding technique, that is traditional plant breeding by crossing different varieties. Okay, high yielding varieties, these are all high yielding varieties. And at the same time, some plant varieties were developed related to disease resistance. Disease resistance by themselves, they can resist to the diseases. Nowadays we are observing no. Okay, some plant varieties they can themselves they can protect from different diseases by the virus, bacteria, or fungal infections. Okay, so that's okay, we can see here wide range of fungal, bacterial, and viral pathogens affect the yield of cultivated crop species, especially in tropical climates. So these disease causing organisms or microorganisms are not only causing infection in animals and human, they are also causing infections to plant species, especially these plants, and reducing the yield. So crop losses can often be significant, up to 20 to 30%, or sometimes even total. Okay, because of this microorganisms. Okay, the crop production was reduced by 20 to 30%, or even sometimes the total crop might be lost for that season because of these disease. Okay, that is it cannot be recovered even by using a certain pesticides or something like that. Okay, we are unable to recover the crop plant. As a result, the total crop will be lost for that season. Okay, if they are recovered, okay, we are lost by 20 to 30%. So that's much effect we are observing with the infections with this fungal, bacterial, and viral pathogens. So it is very, very important to develop a disease resistant varieties of plants. So okay, in this situation, breeding and development of cultivars resistant to disease enhances food production. So in this case, because we are losing by 20 to 30%, or sometimes we are losing total crop also. So it is very, very important. Okay, development of high yielding varieties is very important. And along with that, the development of disease resistance varieties is also very, very important. So that we can increase the food production further. We can cover up those losses. Okay, this also helps to reduce the dependence on use of fungicides and bactericides. So, okay, if you have developed that disease resistant varieties, then okay, the farmer, there is no, okay, the farmer, for the farmer, there is no need of using these chemical pesticides to protect the crop from infections. That means the cost of production is reduced and the usage of chemicals is also reduced by using these disease resistant varieties. So resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constitution of the host plant. So here what we have to do means we have to develop a disease resistance variety, okay, disease resistant varieties. So here we have to develop a resistance in that host plant. Okay, we have to increase the resistance of the, and it is determined genetically. So we have to introduce certain genes into that plant variety by crossing with the wild varieties which are showing the disease resistance. So we have to undergo, that is the resistance of the host is the ability to prevent the pathogen from causing disease and is determined by genetic constitution of the host plant. So before breeding is undertaken, it is important to know about the causative organism and the mode of transmission. So before developing disease resistance in that plant, first of all, we have to understand how the disease is entered into that plant body, either through the roots or through the stem or through the leaves. That is water or through air. Like that, we have to understand. Okay, and the mode of transmission and how it is getting transmitted from one plant, one infected plant to the healthy plant, through air, through water, or through insects, or through the articles that are used during the cultivation, because during plogging, because in between, okay, even after growing the crops, okay, the farmers used to plog, okay, to increase the aeration in the soil, or to remove the, okay, unwanted plants that are growing along with the crop plant, that is weeds. So during that, they use different implements, and through that also the disease might get transmitted. So how the disease is causing and how it is from one plant to another plant, that all we have to understand. Okay, and some of the diseases caused by fungi are rust. Rust, that means like a brown color or black color mass growing on the surface of the plants because of the fungal infection. Okay, example, brown rust of wheat. Brown rust of wheat. Okay, that means okay, either the stem or the leaf. On the surface of the stem or the leaf, we can found a brown color patches developing because of the infection of fungus. And next, red rot of sugarcane. Okay, sometimes when you are eating, okay, if anybody is eating the sugarcane, okay, sometimes in between we'll find okay, brown or okay, brown color patches like structures we develop were observed. Okay, while you are eating the sugarcane, that is a red rot. Okay, red rot of sugarcane. And next, and the late blight of potato. Okay, late blight of potato, that is the leaves will turn into, okay, okay, will form, okay, okay, on the leaves will find brown color or yellow color patches. Okay, that is like, okay, which we call it as late blight of potato. Okay, that is. And next, there are some diseases which are caused by bacteria, like a black rot of crucifers. Crucifers means like cabbage, cauliflower. Okay, rot of crucifers. And there are some viruses which cause infections like tobacco mosaic virus, turnip mosaic virus. Okay, which will cause the tobacco mosaic disease and the turnip mosaic disease, cauliflower mosaic disease. Okay, like mosaic like appearance, that is because the infection with the virus, the cells will undergo death. As a result, okay, that is called necrosis. As a result, we can observe some patches are developing which appears like mosaic like appearance. That's why we are calling it as mosaic. Okay, which is caused by the viral infection like tobacco mosaic virus, turnip mosaic virus, cauliflower mosaic virus. Okay, okay, that is how, okay, that is. And next, the different types of diseases we can observe with reference to plant species or crop varieties. Okay, and we have followed different methods to develop this disease resistance variety, that is by conventional breeding, mutational breeding. Like that, we have used different techniques for development of this disease resistant plant varieties or crop varieties. So first we'll go with the conventional method and later we'll go with the mutational breeding. So methods of breeding for disease resistance. Okay, breeding is carried out by the conventional breeding technique or by mutation breeding. So by developing a plant by using certain chemicals or exposing to certain okay rays like X-rays or UV rays. Okay, we are introducing certain genes or we are undergoing certain changes in the genes and we are developing some new genes which can okay provide an resistance to certain diseases. That is by mutation breeding. And second one, okay, first one is conventional breeding. That is already certain wild varieties are there which are naturally showing certain disease resistance. Okay, in that certain genes are present which are showing that resistance, and those are crossed with uh, the crop, okay, the crop varieties, and to introduce that disease resistant gene into that crop variety. Okay, that is conventional breeding. So we are using both conventional breeding technique and mutational breeding depending upon the type of the plant variety. So okay, the conventional breeding technique is as usual the same as that of high yielding variety. The method is same. So first we'll do one high yielding variety and a wild variety which is showing disease resistance. Okay, and we'll undergo evaluation and will obtain the pure lines. Okay, selection of parents. Okay, evaluation and selection of parents, and later they are undergone cross hybridization and the hybrids are obtained. The hybrids that are obtained, okay, the hybrids that are obtained are tested, okay, tested in the scientific fields and later by the okay in the crop fields, and finally they are released into the market. So the method is same, but the characters that we are selecting is different. Okay, that is what. So conventional breeding technique described earlier, as we have described earlier, the method. Okay, but for high yielding variety, whatever the procedure that we have followed for high yielding variety, the similar procedure will follow for this disease resistance to develop disease resistant varieties. So that is the conventional method of breeding for disease resistance is that of hybridization and selection. So first of all, selection is important. Selection of the parents and followed by hybridization. After hybridization, selection of the hybrids is very okay. These are the three key process that we have to follow while we are undergoing conventional breeding technique. So its steps are essentially identical to those for breeding for any other agronomic characters such as high yield. So that means okay, whatever the plant varieties that we are developing, either high yielding variety or to improve the quality, okay, to develop to develop new varieties which can grow in different environmental conditions. Okay, whatever the agronomic characters we are developing in the new variety by using this conventional breeding, in the same manner we can also follow the same technique for a disease resistance also. Okay, the same. Okay, first germplasm collection, there is a collection of different variants. Okay, and next followed by okay the collection of selection of parents, okay, which plant varieties we are selecting, and next okay followed by the hybridization, cross hybridization. And after cross hybridization, we already said okay, all this will not be successful. Okay, it may be 100 in one or 1000 in one cross will be successfully developed the new variety with the disease resistance and high yielding, and those are carefully selected, tested in different fields, and finally they are released into the market. So that is the technique that we had. So the various sequential steps, that is screening germplasm for disease resistant source, hybridization of selected parents, evaluation of hybrids, and testing and release of new varieties. So the process is the same. Whatever the agronomic characters you want to introduce or you want to develop, okay, we are following the same procedure. We are following the same procedure. Okay, that is so. What are the different plant varieties or crop varieties that were developed by using this conventional plant breeding? Okay, for a disease resistance, that one we'll discuss in the tomorrow class.